Hello everyone and welcome to the second part of lecture 1. We're going to be going over fermentation, gluconeogenesis, as well as the Cori cycle. So recall that we finished our conversation on glycolysis, right? In glycolysis, what we formed was pyruvate, and we also formed these other molecules such as NADH as well as ATP. So typically when we have oxygen in our environment, this NADH is going to go to the electron transport chain that's in the mitochondria, and it's going to donate those electrons into the electron transport chain. Those electrons are going to be shuttled down and interact with the oxygen to create water. So let's say, for example, if we didn't have oxygen, right? So then that means that the ETC won't be working. And by extension, the Krebs cycle and the pyruvate dehydrogenase cycle, which is also in the mitochondria, are also going to stop working. So what can we do to make sure that we still have ATP production, right? Let's say we're in a condition where we don't have oxygen. Let's say we're running a marathon and we're getting really, really tired, and the oxygen in our system is getting very low, right? We have a lot of CO2 building up from respiration. And so what can we do in this condition where we don't have oxygen? Or maybe you're just a species, like not a human, let's just say you're a yeast cell, for example, and you're not in a condition where you have oxygen around. So how can we continue to make ATP? So what fermentation does, is what we're going to be talking about today. What fermentation does is that it allows glycolysis to keep occurring. Well, why would glycolysis stop in the first place? Well, look at the products of glycolysis, right? We have NADH occur, we have ATP. ATP is awesome for, when we need energy, but NADH is one of the products, right? So what if we just keep letting glycolysis occur, right, without fermentation? But what would happen is that, maybe we still have glucose. However, we're going to be running out of NAD+. And we start with NAD+, we have a redox reaction, and we reduce NAD+ into NADH. Okay, so if all we have is NADH, then how can we keep glycolysis happening if we don't have NAD+? So what fermentation does is it simply regenerates that NAD+, right? So we have two different types of fermentation. We have lactic acid fermentation and alcohol fermentation. So lactic acid fermentation, again, just in an anaerobic condition, the ETC is not functioning, thus by extension, the PDC and the Krebs cycle is not functioning. We're going to be regenerating our electron transporter, the NAD+, and then we're going to have this process occur in the cytoplasm. And the reason why it's in the cytoplasm, an easy way to remember it, is because glycolysis is also in the cytoplasm. So we want those two reactions to be really close to each other in case an emergency happens and we don't have any oxygen, right? Excuse me. I'm so sorry, guys. This has been, I just had a really big meal. Sorry. Okay, so so let's say we continue with our conversation with lactic acid fermentation. So this would be the mechanism. So we have our pyruvate, the product from our from glycolysis. We're going to have the NADH that was also another product from our glycolysis. We're going to take the hydrogen that's on our NADH and the hydrogen that's free floating, and we're going to reduce. So we're going to give those hydrogens, or give the electrons. Remember, reduction is giving the hydrogens or giving the electrons, or reducing bonds to oxygen. So in this case, we're going to be donating hydrogens to our pyruvate. The hydrogens are going to go to this carbonyl group right here, and it's going to reduce it to the OH group, forming lactic acid. So just to summarize, the products of lactic acid fermentation is NADH and lactic acid. And just a quick note, we can't keep having lactic acid, this process occurring, because eventually we're going to have a buildup of lactic acid, and that's actually poisonous in high concentrations. And that's why typically we just stopped, what's it called? We become very, very tired after a lot of exercise because of the effects of lactic acid and the effects of glycogen being running out in our muscles, and there's a lot of factors. But okay, our next conversation is going to be on alcohol fermentation. Right? Before we had lactic acid, this is the alcohol fermentation. This is something that we typically find in yeast cells. Right? Alcohol fermentation happens in other organisms as well, but the typical species that we look at when we analyze it is yeast. Okay, so what is yeast? Just know that it is a single cellular fungi eukaryote. So remember, fungus is also underneath the umbrella term of eukaryote. Just like us humans, it's just a single-celled eukaryote. That's something you don't see very often, a single-celled eukaryote. That's what a yeast is. And basically the yeast is under the same conditions as us, right? It just doesn't have any oxygen and wants to keep producing ATP from the oxidation of glucose. So it's going to make sure it has the ability to continue those redox reactions by regenerating the NAD+. A side fact, if you ever tasted something called beer or wine, it is these yeast cells that are producing the ethanol or the alcohol that is found in your beverage. They just put them into big vats, and then they allow fermentation to happen. They take out all the oxygen from there, and the ethanol comes pouring out. Again, even for the yeasts, their fermentation products are deadly to them in high concentrations. Okay, so here's the production, or sorry, the mechanism of alcohol fermentation. So we have here pyruvate, the product of glycolysis, and we have a hydrogen or a proton come in and is going to what's it called? It's going to replace here. I need to draw this actually. I just remembered. So what's going to happen is the carboxylic acid group right here is going to rotate off. Right, we're going to have this bond form a double bond rather than have a single bond between these two carbons. We're going to have the double bond occur between the carbon and the oxygen. We're going to have this form, carbon dioxide, and the hydrogen is going to come replace that bond that was initially bonded to the carbon, and is going to make an aldehyde group, right? So we have acetaldehyde. This is a 2-carbon group, and then our NADH with another proton is going to reduce this carbonyl group until we have an OH group, which is going to be our ethanol. Remember, ethanol, if you guys were remembering organic chemistry, ethanol is just, remember, F has 2 carbons, and then all means a alcohol group. So that's basically what we have here, and that's just another way to represent it. So let's continue forward. Okay. All right, so next we're going to have the conversation of gluconeogenesis. It's just a pathway that, as it says in the name, is going to produce glucose, right? Glucose. Neo means new, and genesis means to start. So we're going to be creating new glucose. Okay, this process occurs in the cytoplasm, but it occurs in the cytoplasm specifically of liver cells. And the reason why we can explain why it's in liver cells is that the liver is going to be the organ that really is going to be taking care of the homeostatic control of your blood glucose. So you have blood glucose, maybe you have a lot of blood glucose, there's hyperglycemia. Maybe you don't have a lot of glucose in your blood, and that's hypoglycemia. Those are 2 conditions that are not good, and so your liver is going to be helping control that through different pathways such as gluconeogenesis, as well as glycolysis. That's something we talked about in a different lecture. So the gluconeogenesis is going to occur in the liver. Basically, you don't have a lot of glucose in your blood. Let's say you didn't eat for a while, and your liver is like, we're going to need to get some sugar in the blood, or else we're going to be hypoglycemic. So what it does is just it takes random, not random, but typically things that you don't find involved with the production of glucose. For example, there could be Krebs cycle intermediates, or you can have carbon skeletons of amino acids, or we can have your well-known pyruvate or lactate also participate in this process. So you're just going to take intermediates found from different pathways, or maybe the same pathway, and you're going to turn those through a bunch of reduction and oxidation reactions back into glucose. So this happens, this pathway happens in the cytoplasm, right next to the membrane of the cell. Remember, if we want a lot of glucose back into our bloodstream, then we're going to want it to be really close to the cell membrane. And so the process occurs in the cytoplasm, just so that the glucose can easily navigate into the bloodstream outside of the cell, and continue forward and increase the concentration of blood glucose. So it's going to occur in the liver, as we discussed, and it's going to require 4 ATP, 2 NADHs, and 2 GTP. So this is a really costly process, right? But at the same time, being hypoglycemic isn't good. So your liver is going to create glucose to make sure it can have a proper amount of concentration of it in the blood. So it's going to cost, remember, GTP is just going to be another, basically something that you find that's equivalent to 40 ATP. So basically we have 6 ATP and 2 NADHs that's going to cost to make one glucose molecule. What's kind of fun and funny and convenient about gluconeogenesis is that the same enzymes that you find in glycolysis is also going to be used in gluconeogenesis. However, there are some enzymes that we discussed that are irreversible. For example, hexokinase was irreversible. The pyruvate kinase was another one that was irreversible. For those kinds of enzymes, we're going to replace them with new enzymes in gluconeogenesis. But for the most part, if the process was reversible, like for example, enolase, that was a process that was reversible. Well, if gluconeogenesis is the production of glucose and glycolysis is the catabolism of glucose, then we're going to have basically the same enzymes being involved in that process. So you just have to reverse some things. Okay, just the next things to note, if you're going to be using these things to get gluconeogenesis started and completed, you're going to be producing these at the end, right? Just removing a phosphate from your ATP, making ADP, removing the hydrogens and oxidizing your NADH back into NAD+, and removing a phosphate from your DCP to make an GDP. Okay, let's go into the mechanism of the cell. So the mechanism of the reaction. So here we're going to start off, for example, let's say with our pyruvate. It doesn't necessarily have to be pyruvate. We can jump and start from oxaloacetate, and as we mentioned before, there's some amino acid carbon groups that can be interpreted, or sorry, not interpreted, that can be inserted into this mechanism. But we'll get into that when we talk about protein catabolism. Okay, here we have your pyruvate, which, remember the structure of this molecule. We're going to have a pyruvate carboxylase, and carboxylase is going to add a CO2 group. So when you have a decarboxylase, is removal of CO2. Here we have carboxylase, which is the addition of CO2. The CO2 combining with this molecule, it's going to take energy, right? So you have a random CO2 is typically found as a really small structure. So the enzyme is going to have to catch the CO2, not only catch it, but also put it onto the pyruvate. So that's going to cost some energy. It's going to take an ATP. Then you have oxaloacetate form, where you just have your CO2 come in and bind, and oxaloacetate. And then oxaloacetate is what's going to be participating in the gluconeogenesis. However, oxaloacetate isn't able to leave the mitochondrial matrix. It has to be in the form of L-malate, or just not oxaloacetate, in order for it to leave. Oh, I don't think I have it labeled over here. L-malate. My bad, guys. But I think I have it labeled down here, right? Yes, perfect. Malate. Just put the L there. Another blunder. Geez. Okay, let's save that. Yeah, so we have an L-malate forming over here. Do I have it in my notes? Let's be converted to malate first. Perfect. Okay. Good catch, Brian. Okay, so we have the oxaloacetate forming to L-malate. It leaves the cell, and after that, it turns back into oxaloacetate. The malate dehydrogenase is going to be facilitating that process, right? Oxaloacetate to L-malate via malate dehydrogenase, and because it's reversible, L-malate is going to go back into oxaloacetate. Next, once it's back into the cytoplasm, we're going to have the oxaloacetate be catalyzed. It's going to undergo a reaction under Pt.2. Pt.2 is just going to take a GTP molecule and add a phosphate group to the molecule, excuse me, and remove that carbon dioxide group that the pyruvate carboxylase got. So it went from a 3-carbon structure to a 4-carbon structure, and then back to another 3-carbon structure, right? So remember, glucose is a 6-carbon structure, and for that reason, we're going to need two of these reactions. Excuse me. We're going to need 2 of these reactions, and that's why we'll get into the products later. But we're going to have a Pt.2, just add a double bond to our group here in the oxaloacetate, also remove a CO2, getting a PEP molecule. Okay, and next our So the names of these molecules are found in glycolysis, and that's why they're all abbreviated here. So phosphoenol pyruvate is the name of this molecule. Phosphoenol pyruvate, that's also found in over here, right? PEP, phosphoenol pyruvate. So if you ever get, okay, like what is that name again? All of the names are here, and that's why I have it abbreviated in the gluconeogenesis pathway, because it's a rather long pathway, and I didn't want to like write all the names and get it really kind of like cluttered. Okay, so we have the PEP, or the phosphoenol pyruvate, come in and now interact with the enolase enzyme. Remember, enolase is going to, an enol group is a double bond, and then in alcohol, in this case we have the OH, B, and OP. And enolase is going to remove that enol group, so you're going to have no more double bond, have a 2-phospho, I think it's a glycerate. Yeah, okay. Who wants to bet? 2-phosphoglycerate. Let's go. Yes, 2-phosphoglycerate. Awesome. And the reason why I was able to determine that was because I remembered the structure of the molecule, that it has a carboxylic acid group here at the end, right? So like, you know, you guys asked me, I think earlier, yesterday, or actually today, if like we should remember the structures, and I really think that knowing the structures can help you out a lot, because I forgot what the G meant, but because I knew what the structure looked like, I was like, okay, that's a carboxylic acid group. We have a phosphate on the 2. Okay, it has to be a 2-phosphoglycerate. Right? So I'd really suggest knowing the structures. But again, I mean, if you're trying to like just memorize it, I think that's fine as well, because you know there's a bunch of stuff to go over. If you don't have the time to know the structures, you know, you don't have to memorize them. But I would highly suggest you simply just to make the memorization process go faster. Okay, next we have the PG mutase. So the phosphoglycerate is going to undergo a mutase. So just going to mutate, just make an isomer, moving a phosphate group down to the 3rd carbon. You're going to have a phosphoglycerate carboxylase, or sorry, phosphoglycerate kinase come in and take a phosphate from your ATP, making it ADP, and giving a second phosphate, creating 1,3-biphosphoglycerate. After that, you're going to have the glyceraldehyde 3-phosphate dehydrogenase kind of reduce our molecule. Remember, the NADH is the reduced form, and then we're going to reduce this. And if this thing got reduced, that means that this thing got oxidized. So it did get oxidized to NAD+. We have our glycerate 3-phosphate. Glycerate 3-phosphate is then introduced to fructose bisphosphate aldolase, which is going to take the glyceraldehyde 3-phosphate and our dihydroxyacetone phosphate, and combine those two. Remember, this is a 3-carbon molecule. There's another 3-carbon molecule. You're going to have a fructose 1,6-bisphosphate form. Then fructose 1,6-bisphosphate is going to remove a phosphate group. In my notes for biochemistry, when I took it with Professor Dice, he had that the ADP was coupled with this reaction, and you formed an ATP when this phosphate come off. But in the TPR book, I saw that they didn't have that there, and so I wanted to go for the thing that the MCAT would go with, not what I learned at Rutgers, and so I changed it to a P, as it had in the TPR book. And then we have fructose 6-phosphate, a phosphoglycerate isomerase is going to the PGI phosphoglucose. So that's why the structure is so important, right? If you didn't memorize that, like, so glucose after that. So hopefully I'm right. Give me a second. I need to confirm this. Ooh, yes, phosphoglucose. Awesome. Yes, it's a lot of things to memorize, of course, but knowing the structure can really help you out decipher some things. Okay, phosphoglucose. Awesome. We have our glucose molecule here. Phosphatase is going to be our last enzyme. It's going to remove our last phosphate group, and we have a glucose. Okay, and here in the notes section, we have the inhibitors and the positive regulators of the pathway. He's just going to talk about what regulates glycolysis and what regulates gluconeogenesis. So remember, glycolysis is the lysis of glucose, and that means it's breaking it down, and gluconeogenesis is the creation of new glucose. For that reason, if those two reactions were occurring at the same time, all you would have is just the production of glucose, the catabolism of glucose that's happening the same cell, just a big waste of energy, right? Just building something up just to like break it down. Like that's sort of odd, and for that reason, they're what's it called? They're regulated by the same similar processes. So if something is going to inhibit glycolysis, then that same process is going to activate gluconeogenesis, and vice versa, right? If something is going to activate glycolysis, that same process is going to inhibit gluconeogenesis. And that's something that's called reciprocal control, right? It's just a single molecule, sometimes a single molecule that came from a process, regulates two enzymes in the opposite way. For example, high AMP, so adenine monophosphate in a starved state, so like you haven't eaten it for a long time, you don't have a lot of ATP, increases glucose breakdown, of course, like we want to oxidize glucose and create ATP down to ATP rather than exhaust ATP to create glucose, right? So ATP is used to glucose neogenesis, and for that reason, AMP is going to stimulate glycolysis, but it's going to inhibit gluconeogenesis. This is an example of reciprocal control. And just remember, if we're going to do some sort of regulation, then we're going to do a regulation at an irreversible step, right? The reason being that we wouldn't do it on like one of these steps, right? If we were to inhibit something at the enolase, well, enolase is an enzyme that's found in both pathways, right? Both gluconeogenesis and glycolysis uses enolase, but there's certain enzymes that catalyze a irreversible control, irreversible reaction. For example, hexokinase or pyruvate kinase. If you were to inhibit those. Oh, sorry, wrong slide. If you were to inhibit those, then you'd be tackling, or you'd be specifically inhibiting a certain group, a certain pathway. So that's the reason why we wouldn't be regulating a reversible step, right? Because that enzyme in the reversible step is found in both pathways. We don't want to activate both pathways or inhibit both pathways, right? We want either one or the other working at a point in time. So insulin and glucagon, what they do, insulin is going to intake glucose from the bloodstream into the cells, and glucagon is going to push glucose into the bloodstream. So how do these guys regulate glycolysis and gluconeogenesis? Well, what they do is that they stimulate a single enzyme. This enzyme is going to be creating something called a fructose 2,6-bisphosphate. And what fructose 2-bisphosphate does is that it increases or it positively regulates phosphofructokinase. And phosphofructokinase is an enzyme found in glycolysis. And for that reason, due to reciprocal control, the production of fructose 2,6-bisphosphate is going to, if it's going to help PFK, it's going to inhibit fructose 1,6-bisphosphatase, which is found in gluconeogenesis. So kind of think about it as like a brother stabbing another brother in the back. It's just like, oh, we're like very similar in name and structure. I thought you would help me out. And it's just like, nah, man, I rock with phosphofructokinase. Like, I rock with them better. So fructose 2,6-bisphosphate is going to be helping out PFK, helping glycolysis, and is going to be inhibiting fructose 1,6-bisphosphate, an enzyme found in gluconeogenesis. So insulin, okay, which one would insulin, what would insulin do? Insulin would create this, right? And glucagon would destroy this, right? Because if this is destroyed, aka not present, then fructose 1,6-bisphosphate is able to run, and fructose 1,6-bisphosphate for gluconeogenesis. Okay, our last topic for today. So if you have any questions about this, please feel free to shoot me a message. So if we go over some basic rules for regulation, they're not firm rules, but these are just a typical trend that we find. There's general rules that we tend to find. Sorry about that. Okay, so the general rules for regulation. So typically, if we're going to have a irreversible step in our pathway, typically that's going to be the enzyme of that step is going to be the one that is regulated. typically is going to be the enzyme that catalyzes a very exergonic or very spontaneous reaction. Okay, next we have our second rule, which is that intermediates tend to kind of negatively feedback and halt the thing that created them. For example, if we had A, and during our process in A, we created B, let's say there's an enzyme here, well then, B floats off somewhere else and does a B thing. But if too many B's are present, right, then the B's like, okay, we're high in concentration, and the high in concentration B's are going to be interacting with A, and then it's going to halt E, right? It's going to halt the enzyme. So that's called negative feedback inhibition, right? The product is going to inhibit the thing that created the product itself. That's something we constantly or frequently find in biochemistry, that we don't want any waste, right? So if we have too much of something, then this just is going to inhibit, like it in and of itself is going to inhibit the process that made it. That's our general second rule. And our last rule is that for cellular respiration, we find that the ATP or ADP state is going to determine whether a pathway is on or off, right? So if you have a lot of ADP, meaning we have low ATP, if we have a high ATP ratio, remember ratio is just a fraction. So if ADP is high and ATP is low, high, this one's low, then we're going to have a big number, right? So we have a high ratio. And if we have that, that means that the energy is starved, right? So like, oh man, I'm really hungry. I need some ATP. So it's going to, the process that's going to be positively regulated would be like the catabolic process, right? Like glycolysis. But if we have a lot of ATP, on the other hand, if we have a lot of ATP and not a lot of ADP, or if we have a lot of NADH and not a lot of NADP plus, NAD plus, then what's going to happen is it's like, okay, this energy is good to go. It has everything it doesn't need anymore, so let's not be wasteful. And it's going to prefer anabolic reaction, right? Anabolic reactions kind of like gluconeogenesis or glycogen synthesis to like store the glucose, or even fatty acid oxidation is something we're going to get into later on. Okay, so the last cycle that I want to talk to you guys about today is the Cori cycle. It's one of my favorite cycles in biochemistry. Basically, it explains why you're able to keep running although you have a bunch of lactic acid buildup, right? So if you have, let's say for example, you're running a marathon and you're pumping your arms and stuff, and in your muscles you have your glucose, of course, and then if your glucose is all used up and stuff, then well, first, like, where can you get more glucose, and second, how can you get rid of the lactic acid that's building up your muscles because you're not breathing, right? Because you're just really tired and you're like, exhausted. And if you don't have enough oxygen, and if you're running low on glucose, then who's going to save the day? Well, the Cori cycle is going to save the day, right? It's going to remove the lactic acid that's found in your muscle, and it's going to replace that lactic acid with glucose. So what we find is that, remember, our liver is going to be our control for glucose. It's our main site. The pancreas is the one that creates insulin and glucagon, but the liver is where the insulin and glucagon mainly is going to be affecting. So what happens, okay, you have a lot of lactic acid building up, and to remove that lactic acid, it's just shuttled through the Cori cycle. It's not a physical thing. It's something that happens. There's not like a physical, like a Cori protein that does anything. It's called the Cori cycle. What we find in the cycle is that, okay, let's say we start in the muscle, we have glucose, we oxidize it to pyruvate. Okay, we don't have any oxygen because we're pumping our arms, we're really tired, not a lot of oxygen getting to our muscles. So we're going to turn it into a lactate, right? We turn it into lactate so we can get our 2 NADHs and go back and take those 2 NADHs. So basically fermentation, lactate dehydrogenase, and put those 2 NAD pluses that we get from fermentation and put those back into glucose so we can get some more ATP. But those 2 lactate are going to travel, and be found in your liver, right? Remember, what can happen in the liver? Well, gluconeogenesis can happen in the liver, right? So we're going to take those 2 lactate, we're going to oxidize them, right? If we took 2 NADH and turn into 2 NAD plus to make lactate, that means the opposite is going to be true, right? We take now the 2 NAD plus, and we're going to oxidize our lactate using lactate dehydrogenase, and we're going to make 2-pyruvate, right? The hydrogens from the lactate are removed by the NADH plus, and we're going to make from the alcohol group in our lactate to the carbonyl group in our pyruvate, right? And then the pyruvate is going to go into the pathway of gluconeogenesis, and we already know what that looks like. Produce glucose. That glucose is shuttled out from the liver to the bloodstream back into our muscle for it to be used up again. Of course, this is not a perfect cycle, and that's why we eventually get tired and just poop out. We're just like, oh, okay, that's it. That's enough for me today. So that'd be our last cycle. And just remember, guys, this here list, I don't mean to scare you. But yeah, this is just a list of things I would highly recommend that you guys know. Just you could throw them onto an Anki deck and just practice them. They're really good structures to know. I mean, some of them we already talked about, like for example, alpha ketoglutarate we talked about in the Krebs cycle, oxaloacetate in the Krebs cycle, glucose, something we find constantly. These are really good structures and names to know, just in case they pop up on the exam. Okay, everyone, that's it for me today, and I'll see you guys in the morning.